Good morning, everyone. I, Dr. Sakshi Bhardwaj, on behalf of Elton Technologies, welcome you all in our this subsequent session of Women's Day special webinar series with the topic Impact of Micelles and Reverse Micelles in the Drug Delivery with an In-Silico approach. The significance of drug carrier system in the process of drug development and discovery is well known to all. They are used in drug delivery systems such as to controlled release technology, to prolong in vivo drug actions, okay, decrease the drug metabolism, and reduce the drug toxicity as well. Carriers are also used in design to increase the effectiveness of the drug delivery to the target site where the pharmacological action would be there. Okay, basically, these carriers based drug delivery is an alternative approach to improve the bioavailability of the drugs while at the same time providing the necessary protection to that drug as well. Okay, today in our this masterclass, our guest of honor will be enlightening us with the importance of drug carrier system in the process of drug development and discovery. She will be emphasizing on the application of micellar systems as carrier molecules of drugs, as micelles are clustering of hydrophobic groups of amphiphilic molecules when placed in the aqueous medium. Nevertheless, she will be explaining the potential of reversed micelles as carrier of drugs and their analysis by in silico predictions. It is an honor for me to introduce our speaker of the day, Dr. Jayanthi Ma'am. Ma'am has begun her professional career as a research scholar at CLRI Chennai. Her doctorate is in biophysics, which she received from Madras University, Chennai. She is skilled with separating proteins from tissues and purifying and characterizing them using variety of techniques like CDS, pH electrophoresis, PLC, UV, NMR spectroscopy are name of few. She is well versed in short peptide synthesis, characterization, and analysis of aggregation in both the aqueous and the non-aqueous conditions. Moreover, she held the position of senior lecturer at Satyabhama Institute of Science and Technology, Chennai, where she focused on the computational drug designing for drug carriers in the Department of Bioinformatics. Her interest lies in developing new anti-cancer drugs derived from the natural source. Also, she worked on small synthetic organic molecules. So, without further ado, let's start with our today's masterclass. Over to you, ma'am.
Um, Good morning to everyone. Thank you so much, Sakshi, for introducing me so nicely. And let me talk about myself for a few minutes. I'm basically a biophysicist turned into a bioinformatician. Bioinformatician in the sense, a very initial level, I should say. And I have done some work. My present area of interest is designing drugs in cancer and for cancer treatment and for osteoporosis. And apart from that, this piece of work which I am presenting now is a small one which I have done. And this consists of two parts. Okay, right, that I'll tell in detail. So now, let me start with my work. The title goes as Impact of Micelles or Reverse Micelles in Drug Delivery. So now, why do you need a drug as such totally initially? Initially, when we talk about a drug, the necessity for the drug comes because we have some disease, so we need a curative agent wherein we go for a drug. So when analyzing the cause of the disease, we finally land up or find out it is because of the malfunctioning of a protein that gives the onset of a disease. So now we have to know what proteins are, and this gives the reason for us to understand proteins more. So what are proteins? Proteins are nothing but polymer molecules built basically of amino acid units. And these proteins are involved in various cell functions. And in each protein is associated with a specific cell function. And they are structurally related also. And the various function, for example, they are involved in structural transport and defense mechanisms and many other things to mention. And now, when how do we say that the structure of a protein is more important to its function? Because the protein structure is different. So, how are we going to analyze the structure of a protein? So, protein structure analysis is not that easy because they fold into the protein basically is a macromolecular chain, polymer chain built of amino acids. So this is the primary backbone of the protein or primary structure. So when this chain is being arriving or coming to a compact structure to enable or do its function, this process is called as protein folding. So how does it fold and what are the different forces which are involved in protein folding? The forces which are involved is hydrophobic interaction, intermolecular hydrogen bonding, and van der Waals interaction, and many other long range interactions. The long range interactions, we can look into them. If we are going to look into BioVR visualizer, we have that shows n number of many other long range interactions also present. And then what are the result of this protein folding? When a protein is being folded, what is it? What is the advantage it gets? That is, it develops or it has a hydrophobic core within it, which encloses or which is involved in the ligand binding activities. And then comes unfolding. So is the protein always in the folded structure? I don't think so. So then when it is trying to unfold, there you have the entropy to be increasing. So what is entropy? Entropy is a measure of disorderliness. So when and why does it increase? Because as the protein is trying to come out of its structure, the side chains which are there in the protein, backbone of the protein, are being exposed to the solvent. Solvent is predominantly aqueous medium, and hence this gives rise to more interactions, and hence the entropy increases. So as such, studying the structure of a whole protein is tedious, and hence then how are we going to analyze the structure of a protein is by studying or the sequence which is of interest. So we get the sequence from PDB or so many other databases which is involved in the ligand binding or whatever, and we study that particular thing, and then we extend the result to analyze the protein as such. So the folding, analyzing the protein and folding or structure of the protein as such is very tedious, and hence these peptides offer a unique opportunity to bridge the gap between the theoretical and the experimental understanding of proteins. So now, when the protein is in the folded and the unfolded state, actually, the configurational there's a competition between the two, and the configurational entropy, hydrogen bond formation, solvation, hydrophobic interaction, hydrophobic core formation, ion pair formation, all these things determines the folding rate of the protein, and in turn they determine the stability of the protein. So that's this competition plays an essential role throughout the folding process, and this determines the thermodynamic equilibrium between the folded and the unfolded states of the protein. And obviously, the protein will want to be in its folded or compact structure. So now the peptide folding simulations and experiments characterize the dynamics and molecular mechanisms of the early events of protein folding. So now, what is this is my area of interest, that is drug designing. So to explain this slide, we start anyone can start with a new molecule wherein which can be taken from any database, and the molecule can be of synthetic origin or natural product origin. Choose the molecule and then find out on the disease which you're going to work. Find out the target protein and dock the molecule with the protein, and find out the interactions. What are the different interactions available, and analyze or estimate its binding strength, and then find out the proper drug this fitting with the drug-like properties wherein it has to satisfy the Lipinski's rule of five, and then finally we declare whether the molecule can be used as a drug or not. So this is being shown in this slide, and then comes this one, which shows the important how this is where I'm introducing nanotechnology as a tool to deliver drugs. So this part shows the structure to sequence to structure, and then finding out the different active sites, and then finding out the ligands and then interaction and all these bioinformatic tools here. And then once the drug is finalized, how is it being carried to the specific area of infection or whatever. So here it is the nanotechnology tool which is used to deliver the drug to the desired target. So now we'll see what are the nanoparticles or how nanoparticle is important in drug delivery. So drug delivery and related pharmaceutical developments in nanomedicine are those systems whose size lies within the nanometer scale. If its size is going to exceed the nanometer scale, we cannot use it cannot be called as a nanoparticles or whatever. So the use of nanotechnology predominantly in medicine and more in drug delivery is being increasing rapidly these days. So this image shows the various application of nanoparticles. Apart from drug delivery, it is used in receptor mediated drug delivery, magnetic targeting, cell sorting or cell imaging, magnetic resonance imaging, hypothermal or photothermic therapy. So currently, many nanosubstances are under investigation for drug delivery, and more specifically, they can be found more in cancer therapy, cancer or cancer treatment. Also, apart from this, nanoparticles are being used to reduce the side effects of the drugs when the drugs are being used continuously for in the long run. Obviously, when a drug is being used in the long run, allopathic medicines they show some side effects, and these nanoparticles are being used to reduce the side effects of these drugs. And the reverse micelles and micelles are important carriers which are used in drug delivery these days to treat various dreadful diseases. And obviously, of course, the micelles and reverse micelles also have the size, or their size is confined within the nanoscale, and hence those are being used as delivery agents or carrier vehicles. So now when we talk about micelles, what are micelles? So when micelles is an aggregate of surfactant molecules, surfactant or amphiphilic molecules when dispersed in an aqueous medium. When we think of micelle itself, immediately what comes into our mind is the amphiphilic molecules and the aqueous medium. When these amphiphilic molecules, when a particular concentration of these amphiphilic molecules are placed or kept into a aqueous solution, they tend to aggregate in such a way that the hydrophilic or the head group of the molecules are in touch or are interacting with the water molecules in the aqueous medium, and the hydrophobic tail regions clusters among themselves and they form the inner core of the micelles. So these micelles are generally spherical or elliptical in shape. So these micelles, how do we say, see, when we are introducing the amphiphilic molecules in an aqueous solution, we expect them to be dispersed unevenly as such. So when you're going to increase them, their concentration at one particular concentration, they form this micelle, and that particular concentration wherein these micelles are formed are being referred to as the critical micellar concentration. So this is the structure of micelle. And now when we talk about reverse micelles, what are reverse micelles? It's entirely exactly the opposite of the micelle. That is, in a micelle, we had the head groups to be in the surrounding or in touch with the and aqueous, interacting with the aqueous medium. Whereas in a reverse micelle, obviously the medium or the external medium will be a non-polar media or some solvent. And in that solvent, when these peptides are being given or introduced, or not peptides, some amphiphilic molecules are being given, placed in that non-polar solvent, they tend to aggregate in such a way that the hydrophobic groups are in touch with the non-polar molecules in the solvent, and the head group or the charged group will tend to form the core of the micelles structure, right? So this is the reverse micelles or inverse micelles. So now, apart from this, generally when we analyze or talk about reverse micelles applications, I'll come to that a little later. So what are the applications of reverse micelles? Reverse micelles are used to carry hydrophilic drugs in the inner core of them, because hydrophilic drugs will be charged and the head group and the core of the reverse micelles are charged. So it will be easier for the hydrophilic drugs to be encapsulated within the core of the reverse micelles. And now I saw a recent article, the antimicrobial peptides are emerging as a new therapeutic class which has very high potential against infectious diseases. since they are less prone to induce resistance. And these antimicrobial peptides are being loaded in reverse micelles and magnetic nanoparticle based reverse micelles, and they act as carriers as well as external magnetic field guides in cancer therapy treatments also. Reverse micelles in nanotechnology aims to improve the therapeutic efficiency of anti-cancer drugs while minimizing their side effects also. This slide shows or consolidates the application of reverse micelles. So this is the structure of the reverse micelle. You have the head group over here and the hydrophobic tails here. And the application, one area of application is membrane systems for protein separation, enzymatic catalysis, studies of changes in protein structure, nanocarriers for food science, carriers of antioxidants, functional agents, on the Newton, etc. And then nanotechnology and nanomaterial synthesis, microreactor and delivery vesicles, bioreceptors immobilization and storage, and then of course our area that is drug delivery systems. So now with this introduction of micelles, let me go into a case study wherein I have taken a micelle peptide, and then we have synthesized the peptide and studied its aggregational behavior in a non-aqueous medium. And then the same peptide has been simulated by using different softwares and finally analyzed by using Discovery Studio. So that is, the work has been done again by using computational means also. So both the work has been published, and synthesis and the wet lab work has been published in the International Journal of Applied Bioengineering in 2010, and the computational work of that has been published in the International Journal of Drug Delivery in 2013. So this is uh, an example of the work with wherein we can show that micelles are being used in drug delivery also. So the structure, the peptide which I have chosen or I have worked upon is dipeptide, and the name of the peptide is isoleucine isoleucine. So the peptide we read it as BOC isoleucine isoleucine NH methyl, where BOC is the head group protection and NH methyl is the tail group protection. So this is the dipeptide which we have synthesized. So So after analysis, synthesizing, we determine the CMC of the critical micellar concentration and then the aggregation. So this slide shows the experimental values which we have calculated in the wet lab. So here, what is this R value? See, in the synthesis, we haven't used any drug there. Instead, when the peptide is being dispersed in the non-aqueous medium, after dispersing it and finding out the CMC, at the CMC value, what we do is after determining the CMC, that is the concentration of the peptide, wherein it is forming a micelle, we add or inject water molecules into the solution. And then what we do is we sonicate the solution. So when we sonicate the solution, the whole solution, the system is being disturbed, and after disturbing it for a while, and then when we leave it to cool down, and then we again study the CMC values. So this concentration is being called as R value, which is the value of the water concentration to the peptide concentration. So for different R values, we have determined the CMC of the peptide. And for each CMC, we have determined the aggregation number also. So these are the aggregation number. And when we go in for the computational value, we have taken the aggregation number as rounded up one, that is as 20. Instead of 19.6, we take it as 20, and instead of 56.6, we have taken it as 57, and here as 52, and here as 50. This is the value which we have taken for computational work. So the steps wherein this work has been done in the computational mode is, it starts with ChemSketch. So the dipeptide was constructed by using the structural mode of ChemSketch. It was done using ChemSketch, and then it was the 3D structure, and then the Smiles notation was generated, and used the Smiles notation was transferred to the Chem database, and from there we retrieved the 3D structure from the Chem database by using the semi to depict mode. And after having the structure, we took it to the pack mold. We used the pack mold package, and in that package, we had to do so many changes in the programming as to when we construct the reverse micelle for example, in this slide, for the initial first one is aggregation number is 20, right? So here we have to add 20 monomers of the peptide. So when we add the 20 monomers and we have to bring them to a micelle, we had to do many changes in the programming. So wherein one of the thing where did is we had to calculate the inner diameter, external diameter, and all those things. The changes in the programming, all these things has been done. And finally, the micelle has been constructed using PackMod. After finishing this, the micelle has been transferred to the taken in the Discovery Studio BioVia, which where Satyabama has the licensed version of this, and we transferred this to Discovery Studio, and there we simulated it, and the energy of the system has been minimized by using Discovery Studio, the steepest descent algorithm of Discovery Studio, and then the drugs of interest has been taken from the drug bank. So this is the description of the work. So we added chloroform molecules in the external media, and within the micelle we added water molecules. So to add the water molecules, we had to find out the inner volume. For all these things, we use online methodologies to find out. And then the initial configuration of the micelle was done using the PackMol package. After the initial configuration of the reverse micelle or inverted micelle, these structures, as I said, was minimized by using Discovery Studio 2.0. This is the version which we have at Satyabama, licensed version in our department, to be more specific. And the constructed micelles were subjected to minimization using the steepest descent method of algorithm in this, and then the energy values were calculated, and the energy which is of interest for us is the free energy of the reverse micelle, right? So now after finishing these are the different aggregates, right? So these show the aggregates. These are the aggregation number. So we framed these are the aggregation aggregates of the micelles. So this is the 20-mer or the micelle with 20 monomers, and this is the micelle with 43 monomers, and this one with 53 monomers, and this 61, 58, 50, and then 40. Here a question rises. See the R value as it is increasing. You can see that the monomers in the reverse micelle is increasing, and after 61 it starts decreasing. So the question arises, why does it decrease over here? The reason is, as the R value is increasing, right? What is R value? R value is water concentration to peptide concentration. So when R value is increasing means the concentration of water molecule is increased. As the water molecule is being increased, the size of the peptide aggregates is increasing. So beyond this 61 aggregate, this one, what happens is the structure is becoming unstabilized. Unstabilization increases or stabilization decreases, wherein it has to let go of some of the monomers so as to form a even more compact, stable structure with 58 monomers, right? And then again, even if further when the R value is increasing, again it comes down, and still it comes down with further increase in the R value, wherein here this has an R value of 10. So this is how the whole thing is being designed and has been done. And after constructing or after initially design framing the reverse micelles by using the PackMol package, it has been transferred to the Discovery Studio, energy minimization, and then energy calculation. And this one is the how aggregation number was determined in the wet lab by using ANSMR. And this shows the free energy values, right? Free energy, right. Free energy of the micelle. So here the free energy is being given. This Discovery Studio gives the free energy in kilocalories per mole, whereas in the experimental value we have calculated by giving it in kilojoules per mole. So the conversion factor is we have to multiply this one by 4.194 or something. So when we do this, we have the free energy in kilojoules. So now this slide shows the free energy of the aggregates when done when calculated in the experimental methodology. And this shows the free energy given or calculated by the Discovery Studio. We can see that sorry, for each aggregate, the value of the calculated 1 by ds goes in very good agreement with the experimental value. As we were really shocked when we saw it is so well correlating with the experimental value. We were so much impressed. We were so happy to see it, right? So for each and everything, we have the same thing. And then comes after this work, we thought, okay, let us incorporate some drug inside the reverse mindset. So to choose the drug, we had to just like that. We cannot choose it, take them randomly. We had to find out the volume of the drug, whether the drug will be can be encapsulated within the aggregate, and the smallest aggregate is the one with 20. So we had to find a drug within which will fit in that particular volume. So we chose these three different drugs: anastrozole, dapsone, and ciprofloxacin. And by these, the structure of these drugs were retrieved from a drug bank. And then this was again introduced, taken in, constructed by using pacmol. And after finishing the initialization work in the pacmol, this was transferred to the Discovery Studio, energy minimized, and again the calculations were done. Again the calculations, these shows the energy, free energy values in kilocalories, and after conversion that's not required, this is the value. And this has been done for ciprofloxacin. And then the next one is, again we did the same work with dapsone. Here also we added all the drug molecules for the 20 mer and then the other aggregates. And for each one, this was done in using pacmol package. And then after finishing with pacmol, we transferred it to ds. Each one we had to do it like that. And then after transferring it to ds, again we minimized, the energy was minimized, and then the free energy values were found out. So next, this table shows the free energy of the reverse micellar system with dapsone molecule, with the drug dapsone. And then the same work was done with another drug by name anastrozole. And this, the image shows the reverse micelle with the drug within them. And the same process as such, it was done, initialization of the drug, and within the reverse micelle was done using pacmol package, and then it was transferred to Discovery Studio, and therein the energy minimization, and then comes the energy calculations. So here, this is the final one. And here the thing is, we have already done the verified the results of Discovery Studio with the wet lab work, which is in very good agreement. And hence we believe that these results are also good, and hence it is really, we were really happy to have this work done. But then we haven't gone in for the next part of synthesizing the dipeptide once again and doing all this work. This is an entirely different project, extensive project to do, and then I couldn't do this. So, wherein we need to synthesize a peptide, we need a fume cupboard, wherein we'll be able to synthesize them, and all those facilities where we didn't have there. So we just dropped that part and we concluded with this part, and we published this work. So as such, as I've already said, the constructed reverse micelles, the aggregation number, and the free energy was shown to be exactly in accordance with the experimental value, and as determined from by the Discovery Studio. And the constructed reverse micelles along with the drugs shows different free energy values, which have to be verified in the experimental method. And also, since the reverse micelles and micelles are of low molecular weight moieties, they can be used as very well used as drug delivery vehicles for cancer and many other dreadful diseases. So, with this, I conclude my work, and I would like to be happy if there are any questions. I couldn't hear you, Sakshi.
Thank you so much, ma'am, for wonderful presentation. And I think we have some questions.
Yeah, please.
Okay, what is the stable form of protein folding and unfolding?
Stable form of a protein folding. See, protein is the folded form of a structure is specific with respect to its function. So, no protein has, we cannot say that this particular form is a stable form. So, it is always in equilibrium, right? When it is in a solution or when it is in the body, metabolism, when it's carrying on with its metabolic activities, it tries to be in its compact form because it doesn't want to get disturbed. So, the inner core, that is why you have the hydrophobic core within the protein to be buried deep inside the protein. You cannot say hydrophobic core on the surface of a protein folding. It's always on the interior. So that when there are some external factors which tries to disturb the protein also, it doesn't get disturbed to that maximum unless it is very drastically disturbed. So you cannot say
Yes,
that
correct.
this is the stable form or anything like that. I don't think so. Anything else, please?
Okay, the next is how magnetic nanoparticles will work.
Magnetic nanoparticles, of course, when it is having, when you say magnetic nanoparticles, they'll have they are directional in their work, right? Their activities or in their thing. Magnet is when you have a magnetic pole, they are more sensitive. So since these are charged particles, they go along with the magnetic direction wherein it is strong and hence it goes along with that direction. And I think that must be the way how it works. Though I haven't gone in detail in that area, but I think I could answer to this level.
Yes, correct. Next one is can you again explain the functional importance of reverse micelles? Or either way we can take it in the
Importance. Yeah.
difference in between the reverse micelles or the micelles.
You want the difference between
Functional
micelles and
difference.
reverse micelles. Functional,
Functional importance.
see,
Yes.
obviously it depends upon the application. Function depends upon the application. See, you know the concept of micelle and reverse micelle. How a micelle is functioning and how a micelle is working and how a reverse micelle is working. Micelle carries a drug which is micelle has the inner core to be hydrophobic. So that is used to carry hydrophobic drugs. That can be used to carry hydrophobic drugs. Whereas reverse micelles can be used to carry only hydrophilic drugs and not hydrophobic drugs because the inner core is charged, right? So, depending upon the application, the function will vary.
You have any other questions? Okay. Thank you so much, ma'am, for answering all these questions. I think we have
Thank you
done
so
with
much.
all the questions and answers. So thank you once again, ma'am, joining us for the today's session. Thank you all for joining with our this webinar, Women's Day special webinar series.
Thank
And
you. Thank you so much.
thank you, ma'am. Thank you, ma'am.
Thank you, Dr. Sakshi. And I should thank Divya. Without mentioning her name, I don't think my talk is complete, right? I should
Yeah,
thank
yes,
her so
definitely, ma'am.
much, right? It was she who gave me the boost to participate in this. And thank you so much,
Okay.
Divya. Thank you so much.
Thank you, ma'am. Thank
See
you,
you.
ma'am.
Bye.
Thank you.
Thank you.